Good morning, and welcome to the 14th Annual ALS Therapy Development Institute Leadership Summit. My name is Carol Hamilton. I am a Senior Director of Development at ALS TDI. I have been here for quite a long time and been to many a summit, and I believe that today might be one of our best. Today we are really focusing on the science that is going on at ALS TDI. You will meet some of our incredible, dedicated scientists and learn about what's happening. One housekeeping item, Wi-Fi. These are scattered amongst the table. Please select the Renaissance Conference Network and use the pass capital ALS 16. There are people scattered around in black polos that have our ALS Therapy Development Institute logos on them. Please, if you need anything, let one of them know. We should have what we need to accommodate people who are living with ALS. There are straws, there are soft foods, there should be space. If you need anything, please ask any of them or certainly me. Welcome to a day of having the opportunity to gather with other stakeholders in this disease, people who are trying to solve it, people who are living with it, family members who care desperately about finding a treatment for this disease. And with that, I'd like to introduce you to someone who is integral to making that happen, our CEO, Dr. Steve Perrin.
Thank you, Carol, and thank you, everybody, for joining us here this morning. Thank you for Ben, Colette, and all of their teams for getting the logistics of this up and running, and all of you here today. I think we have a really exciting day, looking forward to it. I have the easiest part of the day, and probably the funnest part. I have 15 minutes to kind of lay out what the key things are that you should walk out of the room with by the end of the day. So I think in order to set the stage for that, I'll try to do a few analogies that everybody kind of probably has heard about. Hopefully some folks in the room will be happy that I'm mentioning their names, maybe not. But people always ask, why don't we have a cure for ALS? Or even if you want to be a little bit more conservative, why don't we have a treatment that will stop the disease in its tracks right after diagnosis? That's a really hard question to answer, but hopefully you'll get a lot of clues on a few things, how it works, and the fact that we're making incredible progress towards that very challenging goal. But the short answer is that developing drugs for a disease like ALS is really hard, and ALS is a complicated disease. And so I'm going to walk through from 10,000 feet some of the themes that you're going to hear today, and I'll try to tie them in for you. So with a disease like ALS, one of the first things you have to understand is, what is your drug target? We call it a target. What is the thing that you could identify in a person that gets ALS that could cure the disease or stop it in its tracks? So identifying the target in a disease like ALS can be really hard, so let's compare and contrast for 2 seconds. Cystic fibrosis, disease that affects kids, mediated by mutations or a group of mutations in a single gene in the genome. The gene was identified in 1989, 25 years ago. The first effective treatment, not a cure, effective treatment that impacted about 10% of the young kids that get this disease, was only discovered and commercialized 5 years ago, even though we knew the cause, the target, that long ago. So that's an example of a single gene. In ALS, only 10% of the people that get diagnosed have an underlying genetic cause. The first one of those that was identified way back in '93, the SOD1 gene, we have animal models around it and cell-based models. We had replicated in the lab probably 10 years ago. We still don't understand what the mutation in SOD1 does. We know it causes the protein to fold in an unusual way and it's probably toxic. But we did know that if you could eliminate that toxic protein, you probably could slow down the disease. And years ago, we showed in animal models and cell-based systems that that was a true statement. If you could get rid of the gene or knock down its expression, the animals did better. And yet, we didn't have a treatment that could do that. And one of the most exciting and hopeful things that's going on right now is that Biogen here in Boston actually has a program that's bringing an antisense in that targets the SOD1 gene. And initially it might be only for that 2% of people that have mutations in SOD1, but that could be the first really groundbreaking treatment that stops the disease in its tracks for people with a SOD1 mutation. And the reason why it took so long is it took time for the technology to catch up. So the first step is the target. I just described two examples, one where the target is a single gene, one in the case of ALS where 2% of people have that particular target. What about sporadic ALS? That's 90% of folks. We actually don't know what the underlying genetic cause is. There's probably some genetics involved, but also environmental triggers. There you might not know the target. So the next step, step number two, step one was target, step number two is to build a drug. How do you build a drug when you don't really know what the target is? So there you have to try to understand the biology, the pathophysiology, what's going wrong. And ALS is really hard there. I mean, compare and contrast again. Cystic fibrosis gene, single gene, it's a transporter that brings ions in and out of a cell, and if the channel's not working, it throws off the homeostasis. Building the drug to fix that was actually pretty straightforward. Once they got up and running, they made cell-based systems with the mutated channel, and they just screened lots of shapes, lots of small molecules until they found ones that would make the channel work better. And then they optimized those lead scaffolds, if you want to call them that, so that not only did they make the channel work better, but they also, those drugs weren't toxic when they moved towards mice, and bam, they went into the clinic, and all of a sudden we had our first effective treatments there. So this next step, building the drug, is really hard for a disease like sporadic ALS. You have to model the disease, if you will. We use mice, a few different flavors. Some of them look more like ALS than others. We use cell-based systems, which is pretty new. And the key thing, and it's a thing that ALS TDI does really well, it's to integrate those two things. So integrating your target discovery with the downstream tools for drug development is something that a lot of organizations don't do that well. Certainly biotech does it really well. Certainly pharma does it really well. They've got teams that work together to solve those two things. Many academic labs don't, which is fine. They do an unbelievable job in the target identification space, and they make great models. But then tying that to the drug development process can be hard. So when you're hearing Kyle and Theo talk this morning, think about those two first steps, because both of them play critical roles in our team on the target identification piece, which you'll hear a lot about from Kyle, with how we do that with cell-based models of ALS, and then how we transfer those leads into animal models, because that's the next logical step. So let's hypothesize. So there's your first two steps. Pay attention to the target, pay attention to the drug development process. How do you build that great drug that's going to get into the clinic? Let's say you have the best drug going, works awesome in mice. The next challenge is how do you get it to work in the clinical trial setting? So let's do a little bit more compare and contrasting for 1 second, and then I'm get off the stage for you. So in the SOD1 example from Biogen, initially that's pretty easy. You stratify your patient population and only enroll people in the trial that have an SOD1 mutation. That is your target population, because that's what your drug is supposed to do. The other great thing about that example is they know how to measure if the drug is working, because they have assays that they've built that is very exquisitely sensitive to measure that misfolded protein in the fluid in the spinal cord. So when they put the drug into people, they can actually measure if the drug is hitting the target, knocking its levels down, and then seeing if that correlates with the people getting better. It's a perfect setup for drug development, and most often we don't have it. So why do most ALS trials fail? It's because ALS is incredibly heterogeneous. We don't know who's going to be Stephen Hawking at the time of diagnosis and live for a really long time, and we don't know who's going to lose their battle with this awful disease in a year. And that spectrum between exists for everybody. So that makes it really hard to develop clinical trials. And when you see how some of the clinical trials in ALS have failed and what the variables are that cause the failure, sometimes it's the assays we use to measure disease progression. And the biggest variable in the trial at the end of the day is that the data from clinic to clinic is too variable to measure whether the drug worked. And we've seen that in lots of different trials, unfortunately. And companies that have done a great job helping us make that better, Cytokinetics, they led the way in some of the ways that we can try to decrease that noise in clinical trials. Certainly MT Pharma showed it really clearly in their Japanese studies, where their big phase 2 studies with a fairly heterogeneous group of ALS patients, they didn't see their drug worked, but when they looked at the data retrospectively and they identified a much tighter subset, they did a smaller, more focused trial, and boom, they showed a change in efficacy in that third study. So how do we get around it at ALS TDI? Well, we do it with big data, and that's a really funny word to me because big data means something different to everybody. Big data to some people is remembering what their grocery shopping list is when they get to Shaw's. That's big data. For other people, it could be an RNA profiling experiment where there's 100,000 data points. That could be big data. Big data for us in our precision medicine program, well, one of our greatest collaborators, Google, that's been helping us a lot, thought that the data from our precision medicine program was small data. And to Google, it probably still is. But for us, that's been big data. For every person in the program, of which there's probably quite a few of you here, and we can't thank you enough, we've been tracking folks for 3 to 4 years with lots of different outcome measures. We sequence everybody's genome. We have probably 300 genomes at this point. That's billions of data points right there. What are the other genetic factors that might affect sporadic ALS? We've been taking voice recordings monthly from everybody. We didn't even know how to take a voice recording and convert it into data, but we did it because we thought it might be useful, and you're going to hear some exciting stuff about that today, I hope. We asked folks to wear trackers on their wrists and ankle that measure movement by the 100th of a second. Seems like big data to me, monthly measurements for years. Important, and you're going to hear a little bit about that, but probably not quite enough yet to really understand all of that. The next thing that we're just starting to scratch the surface, and again, it kind of ties the target discovery, drug development to making better trials is some of the things you're going to hear about in Kyle's presentation, where we're just scratching the surface of, we made cell lines from a lot of these people. How can we utilize those cell lines to model ALS in a dish as part of that drug development process to understand why ALS is different from one person to another? And can we use all of this big data together to make better algorithms to make trials smaller and faster? Because right now, that is a bottleneck in our ability to find effective treatments and a cure. The trials require too many people, and they take too long. So you're going to hear flavors of all of that today. The 3 things you're going to have to remember, and I'll be here at the end of the day to remind you. How do you get to the target? How do you get to the drug? And how do we make trials more efficient? And if you walk out of the room with showing a lot of hope in all of those things, that's how we're going to break down the frontiers of getting an effective drug for ALS. Thank you.